Transposans. Transposans, frequently referred to as jumping genes, are structurally and genetically discrete sequences of DNA that have the unique ability to move or transpose between the different genetic locations within a cell. They can transfer blocks of genetic material back and forth between the bacterial chromosome and extrachromosomal replicants, such as plasmids. Here is a detailed breakdown of their characteristic structure and significance. Discovery and basic characteristics. Discovery. Transposons were discovered in 1950s by the American geneticist Barbara McClintock during her research on maize genetics. Her groundbreaking work earned her the Nobel Prize in Medicine in 1983. Mode of action, the process by which these elements move, is called transposition. Unlike other genetic recombination process, transposition does not require extensive areas of sequence homology, similarities between the transposon and its destination site. Dependence. Unlike plasmids, transposons are incapable of autonomic existence and must integrate into a host DNA molecule like a chromosome or plasmid to replicate. Structure and types of transposable elements. Transposons all contain the necessary Transposons all contain the necessary genetic information to orchestrate their own transposition, but they vary in complexity. They are generally classified into two main types: insertion sequences, that IS elements we can call insertion sequences or IS elements. These are the simplest transposable elements, typically ranging from 750 to 1600 base pairs in length. They consist of a gene that code for transposase, the enzyme that catalyzes the actual movement, bounded at each end by inverted or repeated sequences that function as a recognition sites for the transposon. Composite transposon. Composite transposon, these are larger and more complex elements ranging 4 to 25 kb in size. In addition to the genes required for movement, they contain a central segment with one or more genes that encode for additional function, such as toxin protection or drug resistance. Like IS elements, their ends consists of inverted repeated sequences, nucleotide sequences that are complementary each other but in reverse order. Because of these repeats, each DNA strand of a composite transposon can form a single stranded loop containing the functional gene supported by a double stranded stem formed by the hydrogen bonding of the inverted repeats. Biological and clinical significance of transposons. Biological and clinical significance of transposons. Transposons are recognized as powerful mediators of evolution and play a crucial role in genetic variation, like mutations by inserting themselves at a wide variety of sites on chromosomal and plasmid DNA. Transposable elements are responsible for bringing about various types of mutations. Spread of antibiotic resistance. Transposons are a major factor in the spread of drug resistance among bacteria. Many of the resistant genes found on R resistance plasmid are actually carried on transposon. This allows an antibiotic resistant gene to easily jump from a chromosome to a plasmid or from one plasmid to another, spreading multiple resistant traits efficiently. Amplifying genetic transfers. Amplifying genetic transfer. Transposons serve as a natural mechanism for amplifying genetic transfer within microorganism, plants, and animals. Because they can be carried between the cells via vectors like plasmids or viruses, they can spread traits, not just from organism to organism, but even across a different species. Natural genetic engineering. So their ability to recognize, so their ability to reorganize DNA, transposon naturally accomplish genetic manipulation that are highly comparable to artificial laboratory or genetic engineering.